We'll probably have more people walking in the next few minutes, but I don't want to delay. We're very lucky tonight to have two speakers who are some of the prominent researchers in CFS in the country, which, you know, here in Salt Lake, I kind of find it odd that we have one of the primary clinical specialists, and now we've got three very knowledgeable researchers. It's kind of a lucky feeling that I have in that regard. Alan and Kathy Light have long research backgrounds that they've explored prior to getting into CFS research, and so we're happy that they had an open mind and a curious mind in finding this of interest. So let me introduce Dr. Light, and will both of you be speaking? You're just here for... Okay. So Dr. Kathy Light will help answer questions. And when we finish, I'll have a short announcement about a group that is going to be formed for people with CFS. So I'll say that until the end.
Thank you. Actually, it's not that much of an accident that Dr. Bateman and I and Kathy are together. It's really evolved. I think I'm going to... Can you turn this one down or off? Okay, good. Yeah, thanks. It really evolved because we have a pain research center there at the University of Utah, in which Dr. Bateman attended, and we attended, and we met each other, and we realized we had common goals in a lot of different areas. And as a result of that meeting, that's why there were the three of us here. My wife and I, Dr. Kathleen Light, and I actually never collaborated on any of our research projects until this one. So, and I won't go into exactly how that all started, but it was working out quite well. That's after 30 years of marriage, by the way, and now it's like 38 or something like 39. Yeah, right. So, yeah, okay. All right, so I'm going to tell you a little bit about something that many of you already know a little bit about. Some of you may have heard me talk here, or my wife talk here several years ago on using gene expression to come up with biomarkers for chronic fatigue syndrome. And I'm going to give you sort of the latest update of exactly where this is at, and then go off on the deep end a little bit and try to tell you a little bit about exactly what these are, because it isn't really immediately apparent from what we have. So first of all, just to introduce the cast of characters here, myself, my wife, Dr. Kathleen Light, Andrea White, who is our exercise scientist who works with us on all the protocols, Dr. Lucinda Bateman, who many of you know, Dr. Ela Singh, who's doing the XMR virus work that I will not be talking about tonight. I wish I could tell you more about that, but you can ask me questions about that later, but I'll tell you that we've been working on it for more than a year now, and we still have nothing that we really can tell you that's definitive, and I really wish I could. It turns out to be a real tough nut to crack. Okay. Last is Ron Hugan, who is our laboratory manager, who really does almost everything that you're going to see. And I may or may not mention that as I go along, but almost all the data you see, he contributed almost entirely to the development of it. Okay, so let's see if everything works here. That's working. And that's working. Okay. Yes, we couldn't have done any of this, actually, without the generous support of several of the private groups as opposed to NIH. NIH has given us some money. We hope to get more money in the future, but most of what has driven the research so far has been support and grants from OFER, the CFIDS Society, and the AFSA, and also from individual donors. We've been very, very fortunate to have that to put into the research I'll show you now. Okay, so most of you know the CDC definition of chronic fatigue syndrome. I'm showing this only to remind you about one aspect of it. You know that it's chronic or remitting, relapsing fatigue lasting 6 months or more that severely impairs function at least 50% of the time is not relieved by rest or recovery. The only important part of that, since you all know this too well, is that fatigue is in the definition. One of the reasons I bring that up is that fatigue is one of the most poorly studied phenomena that we have, and it's very, very poorly poorly understood. Fatigue, it means something different to every researcher that's out there, and I'm going to give you a different definition of fatigue in a minute here that most of you know very well, which is the clinical fatigue that you all experience, and many of you experience at least. It also must have 4 or more of the 8 following symptoms, and again, many of these you'll be familiar with. Impairment of memory or concentration, so we know there's some central nervous system effects. Sore throat, tender lymph nodes, so something going on perhaps with the immune system. Muscle pain, joint pain, unrefreshing sleep, muscle pain being a very prominent phenomena. Change in type of headaches. Symptoms worse at 24 hours after physical exertion. This is one of the ones that we use to our advantage to try to figure out a way to really demonstrate the biological phenomenon that's behind what chronic fatigue syndrome is. And I have that highlighted because that's what we used to actually demonstrate the biomarkers. It's often preceded by a viral infection, and something like 70% in our of the population we've looked at, and several other groups have found, that 70% of chronic fatigue syndrome patients also have fibromyalgia syndrome. Fibromyalgia syndrome has a different definition, however, and that is that it's widespread muscle connective tissue pain, bilateral upper or lower body for at least 3 months, hypersensitivity to pressure at tender points, at least 11 of 18 tender points and other body sites, and the symptoms generally are thought not to be worse after exercise. It's actually not true, as it turns out, but that's what the American College of Rheumatology says. Of course, both of these syndromes are pretty arbitrary. If you look at both the symptoms that I talked about for chronic fatigue syndrome and those that are defined here, I mean, what's 11 out of 18 tender points? Why does it have to be 6 months long? You know, lots of these things are just arbitrary. They just set there, and yet there's still an argument. There's an argument again and again that we're not looking at a consistent population. Well, with chronic fatigue syndrome, it's really hard right now to really look at the same a very consistent homogeneous population, because we really don't know what all the concomitants of fatigue are. We don't really understand the phenomena well enough to be able to do that. You can try to do that, and doing that you might screen out a bunch of people, or you can include them all and then dilute your results. So it's a real problem, which is one of the reasons we need a biomarker, something that's an objective marker that can help discriminate the different types of fatigue that people have. Okay. Okay, this is just a little graph that shows you some of our recent results, and that's going to be in the paper that I'm working on right now. This is a graph of the mental fatigue before, during, and after exercise. Now, this particular exercise task is happening here, just after baseline, mid-exercise, and then this is after the exercise is over. This is 30 minutes after the exercise is over right here. So these time points are during the exercise. It's 25 minutes of very moderate exercise. This exercise, it doesn't mean anything to you, but it's 70% of the predicted maximal heart rate. What is that? For ordinary people, that's a brisk walk down the hall for 25 minutes. Not even a brisk walk. It's a moderate walk down the hall. You don't raise a sweat. You don't really. Most people, even if they're out of shape, could do this for several hours with nothing going on. This is not true for patients with chronic fatigue syndrome. Okay, so this is what happens to the controls right here for mental fatigue. Nothing. They have virtually none to start with. The exercise doesn't do anything to it. Okay, and this is after 30 minutes after 8 hours, 24, 48 hours after this moderate exercise. These are the chronic fatigue patients up here. These are fibromyalgia patients right here. So, and as you can see, during the exercise, these patients experience an increase in the amount of mental fatigue that they report, and it continues to go up following the exercise all the way out to 48 hours. For patients who have both chronic fatigue and fibromyalgia, this group has chronic fatigue, doesn't meet the criteria for fibromyalgia, but very often they'll have many of the tender points, just not enough to make the tender point criterion for fibromyalgia. These are patients that have only fibromyalgia and do not make the criteria for chronic fatigue syndrome. And you see that during the exercise, they really don't have a problem. But after exercise, they too also show increases in the mental fatigue. This is physical fatigue. This is how tired your muscles feel. And again, all of these patients, all the fibromyalgia, chronic fatigue syndrome only, and those that have both chronic fatigue and fibromyalgia, experience worsened symptoms during the exercise and for up to 48 hours afterward. They have not come back down to the baseline here during that time. The controls, some of the controls, particularly the least fit controls, do experience a little bit of fatigue during, but not much compared to even the baseline up here. A little bit of fatigue during the exercise, but in fact it was right back down to baseline levels immediately after the exercise. So again, it's not a problem for controls at all. Yeah.
Is that self-identified fatigue?
This is self-identified fatigue. We ask them how much fatigue you have, and we ask them to rate it on a scale of 1 to 100. Okay, so yes. Now, and so we didn't give them the definition of fatigue. We just say, what's your fatigue? We let them come up with the definition. It's interesting. Most people know exactly what it is. Even the controls know exactly what it is, and they rate it very well, and the repeat for this is actually just as good as any pain measure. It's the pain, which is next. These are the pain measurements. So again, the controls, really no pain at all during the exercise. The chronic fatigue syndrome patients have considerably more pain to start with. Even though that's not part of their definition, is chronic fatigue, in that this particular group is supposed to not have the muscle pain because they don't meet criterion for fibromyalgia, but they still have more pain, okay? Again, so the arbitrary definition of what is fibromyalgia, what's chronic fatigue, and all these kind of things, that shows us some of the arbitrariness of these definitions. But you see that it does get worse following the exercise, and it continues to get worse up to 24 hours, maybe drops off a little bit, never comes back down to baseline. For the fibromyalgia patients, it's not too surprising. They have more pain to start with because that's part of their definition, but they also continue to get worse, even though the American College of Rheumatology says they don't get worse following exercise. They do get worse. They have more pain. The chronic fatigue with fibromyalgia, they are the worst of all. They start with a lot more pain, and it gets worse and worse and worse. Okay, so this is just to give you an idea that we can actually scale this, and that it looks, this gives us a pretty good impression of what's the difference between a patient with chronic fatigue syndrome, fibromyalgia, and control patients, okay? Exactly what they're experiencing. And I think this is something you all, any of you who have chronic fatigue syndrome, you know this is what happens. Any of you who live with somebody who has chronic fatigue syndrome and don't believe that those people, this is what happens, okay? Okay, the problem is that because the major symptoms here for both of these syndromes, fatigue and muscle pain, the problem is both of these symptoms are subjective. What do I mean by subjective? It's something that only you know you really have. You can tell somebody else you have it, but there's nothing by looking at that person that definitely identifies that you have it. We don't have a blood test that measures it right now. We don't have any other way of knowing other than the fact that they tell you. That's what it means to be subjective. So the question then is how to objectively determine chronic fatigue syndrome and fibromyalgia syndrome. How can we make this objective as something that if you looked on a piece of paper, came off a graph from some measure, that people would say, you've got chronic fatigue syndrome, you have fibromyalgia syndrome. And we determined early on that a possible solution was to use gene expression profiling of genes related to the symptoms of fatigue, muscle, and pain, and immune dysfunction. So we picked genes that we knew were related to the final symptom outcome. So we wanted to try to find the genes that were basically making the proteins that were detecting fatigue. We wanted to find the protein receptors that were sending a signal to the brain saying, I'm very, very tired, don't go on, don't make any more movements. We wanted to find the gene that was responsible for the signaling of muscle pain. We wanted to find genes that were related to immune dysfunction. So we concentrated on trying to find what those genes were. And I won't go into exactly how we did all that, but we did. And we used mouse models actually initially to try to determine what those genes were. And also had some very good friends out there in the scientific community who were interested in determining what muscle pain was, and as a result of that, they really did find out what some of those molecular receptors were. And some of those are listed over here now. So this is a list now. I'm going to show you the biomarker. So the biomarkers that we came up with were combinations of genes. So it turned out that single proteins single genes, like just this one, this is the designation, the scientific designation is ASIC, ASIC, which means acid sensing ion channel. This is a receptor that detects pH, so if your blood becomes acid, it can detect that. But it turns out it doesn't work by itself. All by itself, it doesn't do much of anything. It requires one of these two receptors. This is a P2X, this is a P2X5. These two different receptors actually respond to ATP of all things. ATP is energy of life. You may have all heard about that. But it takes one of these in combination with this to actually produce a signal. If you only have this, you wouldn't signal until you were dead because your pH would have to be so low that you would be dead. But with these two, this becomes more sensitive. So it required those together. Also required this receptor, TRPV1. So TRPV1 is actually the receptor for chili peppers. It's the one that makes chili peppers taste hot. It's the essence of hot. What it does in the body, though, the TRPV1 receptor detects heat. It detects how warm the world is around you. And one of the things that it does is it contributes to your thermal set point. So your body core temperature is partly set by this gene because it detects how warm your body is, okay? But it also contributes to the activation of this complex so that when your muscle is producing chemicals, when you're exercising it, it reacts both to the heat, because your muscle also gets warmer, and to some of the chemicals that are produced to enhance the activity of this particular signaling agent again. And because of that, this combination can signal muscle pain, but it also can signal muscle fatigue. We think something similar is going on in the brain, and the brain uses a slightly different mix of these receptors, but we really haven't studied that. There's a lot of evidence that that's the case. So, and what this is signaling then is signaling muscle pain and muscle fatigue, and you need to think about this. So this is a different definition of fatigue then. This is a sensory phenomenon of fatigue. So this is that feeling that when you start upstairs, your legs are getting tired. This is the feeling from your brain that I can't concentrate anymore. I have to shift my attention to something else. It's not the sudden collapse of your brain where you just go unconscious. It's not that at all. It's this overwhelming desire to shift the focus, to not try to concentrate on what you're concentrating on. So that's the phenomena that we're calling fatigue in this case. However, fatigue also means that you can't contract your muscles anymore. So meaning that if I work hard enough, eventually the muscles fail and I can't contract a muscle. It turns out that's what exercise physiologists have been studying for many, many years. Is that related to that sensory phenomenon, the fact that I feel tired? Yes, it is related, but it's not the same thing. They're two different phenomena, but they are mediated partly by the same sensory receptors. There's just a lot of different rules that go on and exactly how that happens. I've got a colleague who's studying that right now, trying to study exactly the other phenomena, why you stop contracting your muscles. And so he's interested in the same sensory neurons we are, but in a very different way. Okay, so let's go on down the list here. So these down here are adrenergic receptors. These all control the amount of vasodilation you have. So they are receptors that are on your blood vessels and can cause your blood vessels to dilate, so they can get more wide open so more blood can go through, or they can constrict, which will keep blood from going to the area that's constricted, but will then divert the flow to other parts of your body. So these receptors can do that. That's the reason we included them. Also, the other reason to include them is that these receptors that are on your muscle, the ones that signal fatigue, they also signal your sympathetic nervous system to contract your blood vessels or to dilate your blood vessels, depending on if you're a working muscle or not. So in fact, these receptors are signaling the system that affects these receptors. And then later on, just as chance would have it, although it makes some sense too, we discovered that these receptors were also found in sensory neurons. So your sensory neurons were responding to the same messages that were causing your blood vessels to dilate too. And that got really complicated. So the system is clearly circular. So this system affects this one, affecting this affects these. So it gets very complicated. These down here are all receptors and cytokine genes. These are your immune system genes down here. Okay, so that's just to sort of give you the players in the field here. These are controls looking at all of these genes during, after 30 minutes after, 8 hours, 24, and 48 hours after that exercise. So this is before the exercise. You can't even see them because I've normalized them to 1. So they're all 1. Here you can see the colors a little bit if you're really up close. The colors are there, but they're not changing. And I did that. We actually adjusted them to make them the same. Following the exercise and controls, some of them go down, some of them go up. 8 hours later, some of them are up. 24 hours later, some of them are up. At 48 hours, most of them are back down. Some of them are still up here a little bit. None of these differences you see here are significantly different from baseline except for this one right here, the beta 1. And exactly why that is, I don't know. But we have some ideas of why that might be. So these are controls. The next thing I want to show you is what goes on in chronic fatigue patients that also have fibromyalgia syndrome. And this is what happens to them 30 minutes after exercise. So you can compare the two. This is a log scale. So in a log scale, that means that changes that occur here are way, way bigger than changes that occur here. So this, in fact, would be here, and so on. So basically, these would be somewhere up in here if it wasn't on a log scale. And I did that so that everything would fit on the page here, because otherwise it wouldn't. So you can see that these genes are increased following the exercise, and these are increased, and these are increased too. So there's an increase in a lot of these. Okay, next, 8 hours later, again, they're up even more, except there's a few differences in there. 24 hours up, 40 hours still up. Okay, so you can see that this is the phenomena you're looking at here fits very, very well with the fatigue and pain that I showed you that the patients reported. This looks like an envelope of exactly what was going on with them. Okay. Down here, then, is what happens to patients that have chronic fatigue syndrome that do not have fibromyalgia syndrome. This group down here. Excuse me, I got that backward. This is chronic fatigue only. Sorry about that. These people do not have comorbid fibromyalgia. I should have known that when I looked at this. And that's just this particular gene. In patients that have both chronic fatigue syndrome and fibromyalgia, you can see in a minute it's not going to matter which one I talked about, other than that one gene. This is the only gene that's different, really, and this one right here. These two genes are different that I'm going to show you. Here's the rest of it. The rest of the picture is this. And as you see, it's pretty much the same in both of these groups. So for that reason, we're beginning to think that, yes, they can be different because these two genes right here, which are this gene, TRPV1, and the ASIC3, because those genes are different, there can be a slight difference in the syndrome, but basically this is actually a fairly homogeneous group. Okay? What's not homogeneous is what I'm going to show you next. So all these say without AE2A decreased patients, without AE2A decreased patients. And I'm showing you that because what we discovered was that there truly is a major subgroup here. And it's 39% of all the chronic fatigue syndrome patients that we examined here. It's almost 40% of the patients. And this is what it looks like. In fact, the genes that go up here at all times here, none of these are significantly different from the controls except this beta 1. The real difference is between this and controls, and I'll show you controls again in a minute. I'll put them up at the top here in a minute, is this gene right here. That gene is the alpha-2A receptor. The alpha-2A receptor is one of the major receptors on your blood vessels and all over the place that actually causes vasoconstriction. It's what keeps your blood pressure high when you begin to stand up. So if you're sitting down and you suddenly stand up, this gene activates, constricts your blood vessels, shunts blood to your brain so you don't pass out. Okay? So it's responsible, that gene is responsible for preventing what we call orthostatic intolerance, the inability to suddenly stand upright, or to come upright sometimes at all, period. And we found that, in this case, 40% of all the CFS-only patients had this gene going down, and 38% of all the chronic fatigue patients with fibromyalgia had this gene going down as well. And you'll see here that that's the only thing that's really going on here, because here's the controls up here. So this is a very, very different patient group, and it's characterized by the fact, let's see if I have it here. Oh, wrong way. Okay. It's characterized by the fact that these patients, 70% of the patients that showed this gene expression pattern with the adrenergic alpha 2A receptor that goes down, decreases with after exercise, 70% of those people have orthostatic intolerance. And only about 20% or less than 20% of the other group have orthostatic intolerance. And we think, in that case, some of that is just we don't know, we haven't got the mix right here for exactly how to look at it, because they may be the same patients here. Or orthostatic intolerance is actually difficult to measure, so it has to be measured at the right time. So it could be that it's just simply that the clinical measurement of the orthostatic intolerance was incorrect. Yes.
Would medication affect the gene expression? I was on
Yes.
the study, and I take Lidoderm for it.
Yep. So medication can actually affect it, and I'm going to get to that maybe toward the end. The way it affects it is that the medications, Cindy actually we found out, she actually told us about this. She actually gave us one patient. She said, I can't figure out what's going on with this patient. There's something, it's a young patient, he's really orthostatic intolerant. What's really going on? And I looked at his gene expression, and she said he's got all the other symptoms of chronic fatigue syndrome. And I looked at it, and the first thing I saw, because I wasn't looking at it on a log scale, I said, there's nothing wrong with this guy. He looks just like a control. And then I looked and saw that all the way along, this particular gene was missing. It was just plain blank. Because I wasn't looking at it on a log scale, there was just a gap there. And then I put it on a log scale, and then I could see the fact it was decreasing very, very dramatically. Because if you don't put it on a log scale, you can't see the decreases very effectively with this. So, and when I recognized that, I said, gee, how many more of these are there? So I went back to all of the ones we looked at and found, yeah, in fact, there was a large percentage of them in each of these cases, a large percentage of these people, and that if you put them all together, this is all of them together, it's a lot of patients, they all showed this same phenomenon. And then that's when we discovered and went back and said, wow, these people are orthostatic intolerant. What do the drugs do for these patients? Actually very little. So the drugs that are typically given, and Cindy has figured out that this particular group works, because of orthostatic intolerance, Lidoderm is actually a drug that actually works for them. It causes real bad side effects, but it actually makes them feel better for a short period of time. So it's not we know it's not the right treatment, but it is a treatment. But a lot of the other drugs that don't work as well on these patients as do on others. For the other patients, some drugs do actually decrease the gene expression. It doesn't take it all the way down, but it knocks it down. And I may show you some of that at the end of the time here if I can get to it. Okay. All right. So what about patients with only fibromyalgia and not much fatigue? This is a larger group of patients in general, although it's a smaller group on our sample. We only looked at 18 of them. What do they show? Well, this is what they show. Remember the controls? They look like controls. There's almost nothing going on. The gene expression just simply didn't change in patients with only fibromyalgia that did not have chronic fatigue syndrome. So we thought, well, gee, that's puzzling. We should be seeing something here. They have muscle pain. We have genes in there that should be encoding muscle pain. So let's look a little more carefully here and see what's going on at baseline. So remember, I adjusted all the baselines to 1 here. I didn't show you how the baselines might be different. So are the genes different? Before we exercise them, are they different? Well, in the chronic fatigue patients, the answer is no. If I look at the averages of all of them, they're very close to the averages of the control. For the fibromyalgia patients, that wasn't true. So for the fibromyalgia patients, here's the P2X4 receptor. This is control. This is the fibromyalgia patient. This is the TRPV1 receptor. This is control. This is for the fibromyalgia patient. This is the IL-10 gene, or cytokine gene. This is the fibromyalgia. This is the control. We found that 3 different genes here were highly statistically different in the fibromyalgia patient. They were higher, much higher than control. So that was a surprise to us. We really didn't anticipate that at all, that this was going to define the difference in fibromyalgia patients. And then we actually thought about it a little bit, and we recognized that this could be for several reasons. It might be part of the reason that that they don't change that much following exercise. They're already up. They got nowhere to go. Okay, so they do change some. They do a little worse, but it's not like the chronic fatigue patients that you saw before. It may be part of the reason that the American College of Pharmacology came up with that idea that they didn't really get worse following exercise, but they're already bad at baseline. It also could be that these people are, and this particular group of patients, because we were told to do this by our sponsors, were not allowed to be on any medications. And so it's possible what we got here was a group of patients that weren't that affected because they could go off all their medications or didn't have to take any medications in order to be in the study. And we lost the patients that were very, very affected. And that may be part of the reason that they have a different profile and don't show the gene increases following exercise. It's also possible that because they're less affected, they're more active. So they're out walking around all the time, like most of you people aren't. So they're actually doing things, and as a result, they're turning their genes up during the day. And so they come in, when they come into the lab, their genes are already up, and therefore they do have less distance to go. They can't go up much more because they're already up, because they are more active to start with. We don't know the answer to that, but for whatever reason, this defines this group and not the chronic fatigue syndrome patients. Okay, so I just gave you all the answers here now. And you're going to, now there's so many questions you had, right? I'm going to go on and actually tell you more about that. It turns out that the gene expression that we look at follows the severity of the disease too. So these are patients with greater severity, these are patients with less severity. So these are the most severely affected patients, patients that are less severely affected, the less still affected, and the least affected patients. And you can see that the gene expression pattern matches that. And these are controls right here. So you can see that even down here, they're above the controls, but there is a sort of a match. So the gene expression does fit with how severe the symptoms are in these patients. Okay. Okay, but what are we really measuring? Gene expression, what is that? And why are we really measuring that? Okay, so this is the real part of the talk here. We'll come back to the other at the very end here. So what is gene expression? How does it differ from genetic causes? What causes change in gene expression? Why is gene expression changed in chronic fatigue syndrome and fibromyalgia syndrome? And how does it help chronic fatigue patients to know their gene expression is abnormal? So these are the real nuts and bolt questions, as opposed to just showing you data there. So first of all, genetics versus gene expression. Genetics is the result of the DNA coding you inherited from your parents. The genetic code of your DNA that you inherit is initially, at least, the same as in all the cells of your body. All your cells have the same DNA coding in it. The differences in the expression of the genes is why you have different tissues like heart, kidney, bones, and brain. So what makes the genes, makes you different is not all the tissues in your body. What makes a bone as opposed to a brain is not the DNA that's there, because the DNA is all the same. It's the amount of expression of that DNA. How much of that DNA is actually made into protein, okay? So that's what makes it different. Okay, the cell nucleus contains the DNA you inherited from your parents, and this is a pretty picture of a cell that I ripped off the internet, and so I apologize to whoever I ripped off from. I should have put the attribution there. So this is the nucleus. This is where the DNA is. DNA is all the squiggly stuff I penciled in here. And this is penciled in DNA, so it's squiggly stuff, and it's really just really long strings, and there are 44 strings that you have tossed around in your nucleus because you have 44 chromosomes, and so each one of these little strings is broken up into a length, and so there are 44 lengths of these strings. They normally unravel all over the place. When the cell divides, they condense into really something you can see much better with a microscope. So normally it's all strung out all over the place. And all these little bins and loops and things, they mean something. Okay, the DNA is what makes up the chromosomes. It's usually stringy with many windings and meanderings around proteins that are located in the nucleus. So this is sort of the cartoon many of you have seen of DNA that's actually composed of a double helix with little cross bands like this that hold it together, and it's a double helix because it's both a helix to start with and it also bends around itself, and it can coalesce into chromosomes that have this sort of a pattern to it. But if you look at here, this is the code. These are the base pairs pair C, A, and that one's T, I believe this is G. So there's only 3 of these, sorry, so it's got to be just a copy. There's only 3 on each of these sides, and they pair with base pairs on the other side here. So this is a T, G, C, A there. Okay, so this is the code. It's just that over and over again in a different order, all the way along this band here that actually defines exactly what kind of protein is going to be made. All right. So it is the code of life. The order of those base pairs is the code of the life. It is the specific order of that code that the bases are in that occur that actually is the code. And this is at A, T, C, G, T, A, T, and these can be in any kind of order, and except that they define, this order defines exactly what protein is going to be made by that DNA. All right. So this is the DNA, and there's those little cross bands. These are the base pairs right here. All right. So genetics is this code, or ordering of base pairs in the DNA. It's what defines all the proteins and more that make up your body. Okay. So that winding, I started showing you the fact that DNA had little twists and turns in it. It was squiggly all over the place. The winding around the nuclear proteins and other special proteins called transcription factors, which are different in all tissue types. The winding is actually part of what defines what part of that DNA is actually going to be expressed, meaning going to be made into proteins. So if parts that are wound tightly around aren't going to be expressed, parts that are less tightly wound are going to be expressed, are going to make proteins, and that's different in all the tissues in your body, and it's why a bone is a bone and your brain's a brain. So, okay. So in order for the DNA to make proteins that make up your body, it must next be transcribed into RNA. So that DNA must go through an intermediate step. It must be basically an RNA copy of that DNA must be made. And that's called transcription, DNA transcription. So the DNA can unzip itself, so the 2 sides of that thing come apart, and then those base pairs are exposed, and then an RNA copy is made on one of those sides along those base pairs. And the RNA copies a short segment of the DNA, and it makes a copy of that code. The RNA copy leaves the cell nucleus and directs the formation of a protein, and using this to code, it copies, using the code, it copies from the DNA. So this is the DNA, this is a cartoon, and the DNA unwinds a little bit here, forms a strand of RNA, and this is the mRNA. The RNA then goes out of the cell nucleus and then actually is bound to the ribosome, and the ribosome then actually attracts in, that's another transfer RNA. The transfer RNA actually has protein peptides on it. These peptides then, because they're bound to the transfer RNA, line themselves up and then form the polypeptide or the protein. So the protein is actually made from the RNA. Now, I go through all of this because this is what we measure. This mRNA, this messenger RNA, the RNA that will come out of the cell and actually start to make proteins. That's what all that gene expression that you saw is. We're measuring the amount of RNA for specific genes, for specific, that's going to make specific proteins. Okay, the DNA code is inherited, but the amount of RNA you made is affected both by the code, so the code affects the amount you make, and it's also affected by your environment. That's why I want to show you the difference between genetics and what we look at, which isn't genetics. It's actually gene expression. It's because the environment can affect it. So the cells can receive a signal. For example, the chemicals you make in your muscle when you're working. So the cells can get that signal. If they get that signal can activate a transcription factor, which is a protein. The protein can then go in and bind to the DNA. And in binding to the DNA, it can either cause that part of the DNA to be more transcribed, so you make more RNA, or less transcribed, so you make less RNA. You can have both positive and negative transcription factors. So what that means is the chemicals that you're making can actually cause a change in transcription. So can a lot of other things. We'll go through some of the other things that can actually change the amount of DNA that's transcribed into RNA, which changes the amount of messenger RNA. So we're looking at the changes in that messenger RNA and our gene expression. So what we're looking at then is not is only partly what the DNA is made of. Because if you've got a certain kind of DNA with a certain coding pattern, you may never be able to make as much RNA as somebody who has a different pattern. Okay, you start with that. You have that to begin with. However, the environment can change it even more than your inherited ability to make the RNA. The environment can change the amount of RNA you make even more than the pattern that's laid down by your inherited DNA. Okay. So if the protein being made is what signals the brain, in this example, that your muscle is tired, then you will sense more muscle fatigue, for example. And this is what we decided to measure. We decided to measure these particular types of RNAs as a biomarker for chronic fatigue syndrome and fibromyalgia. So what we decided to measure was the RNA that makes the proteins that signal muscle fatigue and muscle pain. That's why we picked those particular genes that we looked at to form the biomarkers. So this is the final part. It's the part that tells you that you're fatigued, that tells you that you have muscle pain. This is not the part that caused that signal that changed the RNA. So in fact, we're not looking at the causes. For example, virus might cause this. In fact, these are some of the things that can change it. Viruses can change the amount of RNA. Stress can change the amount of RNA. Pollutants can change the amount of RNA. Your diet can change the amount of RNA. Exercise can change the amount of RNA that you make. Lots of different things can affect the amount of RNA you make. Diseases of various varieties. Flu can change the amount of RNA you make. Viruses of that type as well. So why do we measure RNA as opposed to measuring the proteins? Well, this is sort of the lamppost version of this. RNA is easier to measure than the signaling proteins because it doesn't take much of these proteins to do the signaling. There's so little protein that it's really difficult to measure it. It's actually easier to measure RNA because we have really good tools to do that. And that's what this says basically. Rapid and accurate methods to measure RNA have been perfected in the last 10 years, and in fact in the last 3 or 4 years they've gotten really good. I don't know if any of you follow the cancer literature at all. There is an explosion going on there in terms of looking at gene expression because the tools are really available. It's really easy and much cheaper to do. Okay. So the RNA changes can give us clues as to what causes the body to increase fatigue signaling. The RNA changes can also give us clues to more permanent changes in the winding and regulation of the DNA that can occur with diseases and aging. So I was telling you about transcription factors. Well, they can temporarily change the amount of RNA, but in fact, there are ways to make those changes even more permanent. Some viruses will do that. Some other just consistent activation of transcription factors over time can actually change the regulation of DNA so that permanently you make more of a particular RNA, or permanently you make less, unless something very powerful happens to change that. So there are more permanent ways to. Okay, why do we measure RNA in white blood cells? Why do we use leukocytes? Because I've been telling you about muscle sensory neurons, right? So why are we using leukocytes then in our measurements? Because that's where we get our RNA from. Well, again, under the lamppost, we can obtain white blood cells easy. So it's easier to find your keys looking under the light. Well, it's not really, because your keys may be over there someplace, but and that's the truth. But it's much easier to look at it here. But there's more logic behind it than that. Brain and sensory neurons that innervate muscle don't grow back. Removing them is not advised, so we don't do that. Okay. On the other hand, white blood cells are also signaled by the chemicals made in your muscles, and we knew this beforehand. So we knew that white blood cells, they react to the signal that you get from your muscles, and so they're detecting the same fatigue signals that your brain is detecting. There is a tissue donation site in case you do want to donate your cells, and that's this particular thing. It's the NIDRI resource. I can hardly read it here. Sorry about that. NDRI resource organ donor programs, individual donors, and this is the site. If you just look up under tissue donation for sensory neurons. It's the National Donor Resource Information is what it is, or something like that. Yeah, I don't know what it stands for either. So, but they've actually been sending me emails on a consistent basis telling me they have dorsal ganglion cells that I could buy from them, which are actually, and they're actually quite cheap, and they will provide them in a form that would be useful. Trouble, I can't use them right now because I don't know enough about how to use them yet. So, okay. So again, back to why do we measure RNA in white blood cells? Well, when signaled, the white blood cells make chemicals, cytokines, that cause sensory neurons to become more sensitive. We knew this. So we knew not only did they detect the fatigue, but in fact, they could actually be part of what's making your sensory neurons send more signals. And in fact, the white blood cells are the immune cell function cells, and they can increase fatigue through cytokines from a variety of diseases and trauma. We've known this for a long time. So even before you had chronic fatigue syndrome, when you got the flu, you felt fatigued and you felt muscle pain. Everybody does. And it's because the viruses cause the immune cells to react and make cytokines. Part of the things these cytokines do is they do help you tear up the viruses and other things, so they actually make you better too. But they also signal the sensory neurons that you're having fatigue and pain. So, and which is part of the reason, part of the idea is that actually protects you. When you're sick, you want all your resources to be put into actually putting your immune system in action and not out running around and doing all kinds of things you normally do, because you want to save those resources to attack the disease. So that's, this is part of what white blood cells do. So part of a large group of people think that this may actually be the case in chronic fatigue syndrome, and that in fact the immune system may be instrumental in causing chronic fatigue syndrome. This has been a major hypothesis in the area for quite a while. So the white blood cells could be the primary cause of fatigue in some conditions. We actually believe, given what we've seen in terms of the activation of white blood cells with the exercise stress, that in fact they contribute not as much as many of the groups have thought. That while they can contribute, they're not a major contributor in a lot of these patients. Okay, so why does gene expression change in chronic fatigue syndrome and fibromyalgia? Why would it change at all? Why should it change? We don't know. That's the answer to that. Because there's not a strong genetic influence, not many strong family relationships, we believe that most of the causes of chronic fatigue syndrome and fibromyalgia are environmental. We actually have a collaborator who's worked with us and has found some families in which chronic fatigue syndrome seems to be inherited. Okay, we haven't been able to study that because we've been not able to get additional sources of fundings until just recently. She just recently got a grant, so hopefully we'll get more into that to try to figure out what's going on in that particular family. So some of you may know about Alzheimer's disease, for example. Alzheimer's disease, only a very small percentage of Alzheimer's disease is directly caused by genetic changes, something like less than 2%. Okay, but studying that 2% of people gave us clues as to what's actually causing the disease in all the other people. Okay, and that's what we're sort of hoping here. Even though we think that the familial relationship, the actual inherited chronic fatigue, is rare, if we could study them individually, we may come up with the clues of what's causing it in everybody else. So that's what we're trying to get at there. Okay, we do know that viruses cause gene expression changes in many types of tissues. So viruses are one of the possibilities for what could cause the changes in chronic fatigue syndrome and fibromyalgia syndrome. Particularly Epstein-Barr has been well studied and almost certainly does cause, maybe not all by itself, but at least itself and then something else, because there are many cases where Epstein-Barr virus mononucleosis was that is fatigue, causes fatigue all by itself, and these patients developed that and never got better. Okay, and they've been well studied. Could other kinds of viruses cause this? Most of you probably are aware there's a thing called XMR virus that's been really highly touted as maybe causing it. And as I told you at the very beginning, I'd like to say it did or didn't, and I can't tell you either. It's a very difficult virus to study. We have one of the world's experts right here in Salt Lake working with us to try to do this, Ela Singh, who was one of the people who first identified XMR virus as potentially associated with prostate cancer. And that's where a lot of the research initiated from her efforts. And she's an absolute expert in this area. Very, very careful researcher, and I'm so glad we're working with her. She is just an expert. Okay. Injury and stress can cause gene expression changes, fairly dramatic changes. And toxins, allergens can cause gene expression changes. So it could be that any of the things that we think might be causing it are causing it. It's likely that some of them are, and some of them may be doing it in combination. So it may take a double hit, 2, 3. So it could be a virus and an environmental effect of another variety. So heat, cold stress, something else. We don't know yet. We need better models for this. We need to understand more what fatigue is, the final common pathway. What's really affecting the final common pathway there that's actually producing the symptoms? Not what's upstream, because everybody gets these upstream diseases and they all get better again. With chronic fatigue syndrome, you don't get better again. Okay, so now I'm going to go back to the beginning. I'm almost done. Which is, okay, so now I've got to explain what we measure and why we measure it. Let's go back to the findings again. Okay, the brief outline of the protocol. We do this blood draws, numerical ratings of mental and physical fatigue and pain, 0 to 100. Whole body air-dyne cycles, 70% to age predicted max exercise, 25 minutes. And then we report all these things. We do post-exercise blood draws, 5, 8, 24, and 48 hours. That's the worst part of the whole protocol. I've done it myself as a control. Just getting all these blood draws, particularly when you're needlephobic like I am. It's amazing how many anesthesiologists are needlephobic. It's a real shocker. You'd be surprised. Then we give them all these tests, and if this doesn't fatigue you, nothing will. This actually made me more tired than I've been in quite a while trying to take all these tests. Okay. So we take all these measures, we do the gene expression levels on leukocytes relative to the housekeeping gene, and I showed you where all these receptors are. The sensory receptors, the alpha adrenergic receptors, the cytokine genes. So each of these are particularly, I explained them before, are particularly there because we knew these were sensory, that these were on the sympathetic neurons, and it turns out they're also on the sensory neurons, except even this is on sensory neurons. And then all of these we put on there because of the hypothesis the immune system was involved here. Of all of these, this is the only one. IL-10 was the only one we found was really changed in patients with chronic fatigue syndrome. Again, here's the data again, only a slightly different organization. Controls. Chronic fatigue syndrome and chronic fatigue syndrome patients that are comorbid for fibromyalgia syndrome. This is 34 of the patients right here. These are the ones without that alpha-2A decrease, so these are ones that did not go down. And here you can see, if we put them together, this group looks even better than the data you saw before. And it looks better because this really is a relatively homogeneous group with the exception of this gene and that gene. Okay? So you can see how much greater this is. It's orders of magnitude greater than controls. This is the major subgroup once again. So this is the chronic fatigue syndrome and chronic fatigue syndrome comorbid with fibromyalgia syndrome. So all of those patients, initially I showed them broken into two different groups. It's all a single group here now. And now you can see that it looks even better than it did before because it's actually quite a consistent group if we average it all together. The genes that are different, this is the only gene that's really different here. The alpha-2A was down in this group. And again, 70% of these patients have orthostatic intolerance. This is fibromyalgia syndrome only patients, and here's the controls again. This gene, alpha-2A, is the difference. Okay, again, what about the fibromyalgia again? I showed you this before. Same thing again. Fibromyalgia only, no changes from controls. The changes are in these genes at baseline. Severity ratings, what were those? Well, this is what the severity ratings I showed you before were. With one being fully disabled, these were able to hold part-time jobs and do the majority of self-care activities. And here we go again. The highest gene changes occurred in patients with the highest severity, less so with less affected, less yet with those, and finally the least, with those that were the least affected. Controls. Okay, gene expression is biomarkers. It turns out these are controls right here. Now I just dot blotted them and showed the increases that occur from adding up these genes right here. And these are the most severely affected. These are less severely affected patients, and these are controls. And you can see that there's a pretty large separation here. And if you use just that with no real manipulations in it, you can come up with a biomarker that has a specificity of 94% and a sensitivity of 65%. This is not so good. The 65% right there, we think we can improve that by throwing in some other genes, but we haven't done that yet. This is actually quite good. The specificity of 94% is really good. If you put these two together, according to the gold standards of biomarker industry, this is a good, actually it's a good to very good biomarker. It's not an excellent biomarker, and we're hoping we can make it somewhat better. But this could be available, hopefully, in the not too distant future, as maybe 2-3 years, as something that could be clinically useful. Okay, so are biomarkers useful in treatment? Well, we'll start right here. The AH2A decreases, so we also has the orthostatic intolerance. This group should be treated differently, right there. So if you knew you were a patient that fell into this group, and if you had both orthostatic intolerance and this AH2A decrease, you're in that group, and you should be treated differently. Are the treatments helping? Well, the subjective measures can be misleading. Patients may be getting better but not know it, or they may be getting worse and not know it. Some patients may benefit, but which ones? Which ones would benefit? Well, it's very possible that the biomarkers that we're using, if we can look at it under these conditions, may actually help us determine these things. And we have a study that we're just starting, and hopefully it's going to begin any day now. We're looking at Lyrica. Pfizer's given us a free grant, and we don't have to give them anything for this, a grant to actually study a crossover design where we actually look at patients on Lyrica and patients that are on placebo, and then cross them over so that in the next period, the people who are on Lyrica will get placebo, and the other people will get the Lyrica, and they won't know what they've gotten. And we'll actually look at the gene expression and see whether the gene expression is getting better with treatment with Lyrica. Do existing treatments have effects on fatigue and pain? Well, yeah, we don't know that, actually. So this is data we had given the scale you just saw before for fatigue and pain. This is pregabalin or gabapentin or Lyrica, as the case may be. And this shows you that in our patient population, this is that same pain mid-exercise, pain after exercise, 48 hours after exercise, that in fact, the patients that were on pregabalin did better than patients that were on other treatments. But that doesn't mean anything all by itself, because this was just taking patients that were on it, and they may have been on it for the reason that they were doing better and all that kind of thing. So we need to actually do the real study, but it's possible. Mental fatigue, same thing. So this is probably a bigger surprise than pain, because the drug companies all knew that pain, at least in some patients, got better with pregabalin or gabapentin. What they didn't know is that mental fatigue also got better. Maybe. Okay, do existing treatments affect gene expression? Well, this is the gene expression pattern. These are patients that were not on the pregabalin or gabapentin. These are patients that were on. And notice these genes right here. These are those sympathetic receptor genes. They're not up here in the patients that were on the treatment. Could be that's why it works. That's really just very preliminary. It doesn't mean anything at this point. Could be. So the conclusions. A diagnosis of fibromyalgia and chronic fatigue based on gene expression is possible. The treatments can be suggested and evaluated by gene expression. I don't know why. This was not supposed to disappear. Something happened there. Sorry about that. The cause of chronic fatigue syndrome and fibromyalgia syndrome could be increased sensitivity of fatigue and pain detecting neurons, either sensory or in the brain. Or it could be dysfunction of the sympathetic nervous system, or both. What's this?
Same slide again from before.
Oh, yeah, I'm answering the questions. This is the quiz. What is gene expression? This is the answer. Now we have with the answers. It is the measure of how much transcription of the gene is occurring. We use quantitative measures of RNA to determine that. That's what we're measuring. That's what you're seeing in all the graphs. How does it differ from genetic causes? Genetic causes involve mutations in DNA. Gene expression is changed both by DNA mutations and by the effects of environment, or by environment only. Okay.
Can I just ask a question
Yeah.
there? Is it environment within the cellular level?
It means all levels of environment. The exterior environment can affect the internal environment, which can affect the gene expression in the cell. So it's both. So, did I answer your question? Yeah. It means everything that has something to do with stuff outside of your body that influences your body and all the stuff that's in your body, which is influenced by everything that's outside of your body. How much water you drink, what kind of water you drink, how bad it is, and all that kind of stuff. Yeah. What causes change in gene expression? Both changes in DNA by mutations, inherited or somatic mutations, and environment through transcription factors. So that's the answer right there. Why is gene expression change in chronic fatigue syndrome and fibromyalgia syndrome? At this time, we don't know. Many possibilities include inherited mutations, viruses, stress, injury, and many, many other factors that we just simply don't know. How does it help chronic fatigue patients to know their gene expression is abnormal? First of all, this is objective data showing that there is a biological reason for the symptoms. And many patients have asked me, have told me that this is something they would really like to see. So that's one reason. And in fact, this is, believe it or not, it's a reason for biomarkers in general. If you look at the handbook on biomarkers, it says this is one of the key reasons to have a biomarker of any kind. Second reason, objective assessment of how serious the disease is can be done with these genes. And in the future, we hope that we can predict which drugs would be most useful by using gene expression. In the future, we also hope that we can monitor the therapeutic usefulness of the treatment, so you can actually try the treatment out. If it's showing the gene expression is getting better, even if the patient isn't feeling better, they might feel better after being on that for longer, for example. Okay. Finally, thanks again to offer for the moral and financial support. Without this, we really couldn't do a lot of what we're doing. So, thank you.